So, today we are going to be doing a couple of different things. We are going to be working through our lab that we had started on Friday. We are also going to be going over the second half of our cell transport. So we have already talked about the different parts of the cell membrane, how it relates to selective permeability. We are talking about the different mechanisms, the different processes that the cell takes to get things in and out of the cell. We've talked about passive transport, diffusion gradients, all of those fun things. And now we're going to talk about active transport. So before we get into the actual lab work, first thing I want you to do with your partner, discuss this question. This was on your test that you will be getting back today. So use the diagram to answer the question, what type of cell transport is occurring in the diagram on the right? So take a minute, think about it. Carter, what type of transport is this?
Facilitated diffusion.
Why?
Because that's what my test says.
Okay, but explain to me from what we've learned. How do you know this is facilitated diffusion?
It's facilitating.
Exactly. Facilitating diffusion.
Yeah. It's facilitating.
What is facilitating?
The
Ions.
ATP.
They're going from high contrast to low contrast.
Concentration, you
Yeah,
mean?
that.
So they are going from high concentration to low concentration. So that means that it is passive transport. We also see on the bottom here, if it was active, you would see this little fancy molecule called ATP. So this is not active transport. What are these purple things?
Ribosomes.
No, the proteins.
They are proteins. Yes, you can see protein channel, carrier protein. These are proteins. So this automatically rules out that it is normal diffusion. So this is facilitated diffusion because, as Noah was saying, the particles are facilitated. They are helped out by these proteins. So yes, this type of transport is facilitated diffusion. Yes.
Question.
Yes.
In some of the diagrams that we talked about, those dots are labeled as ions.
Yes. Sometimes, yes.
Sometimes. So they could be ions or proteins depending on where they are in the diagram and what you're using them for?
These are the proteins. These could be ions, these could be food particles, they could be different molecules.
So does that
Yes.
make sense?
So in this case, when we talk about proteins, we are talking about the ones that are embedded in the cell membrane. The different particles, whether it's ions, whether it's food, whether it's oxygen, whether it's water, those are going to be the particles outside of the cell or inside of the cell. Any questions about this question? Like I said, a lot of you got this one wrong, so I did want to review it just so that we could go through and I could kind of correct any potential misconceptions that you guys had. Okay. If not, please have your gummy bear paper out. So I'm going to pause the recording here. We're going to discuss the gummy bear lab, and then you guys are going to go into it. At about 2 o'clock, that's when we will start our active transport notes. Now that we are done with our gummy bear lab for now, like I said, it will be due tomorrow night. If you have not completed it, please complete it over tonight. If you need to meet up with your people, that's fine. But we are starting on active transport. And the one thing that I wanted to remind you of, again, is this concentration gradient. So concentration gradient was that movement of particles from areas of low or from high concentration to areas of low concentration. So it's like that elevator example that I gave on Friday, where someone farts in the elevator, you try to disperse as quickly as possible. So yeah, in this case, we want to go from areas of high concentration, very cramped areas, to areas of low concentration where it is much more spread out. One thing you will notice with active transport is it doesn't follow that rule. So we are going against the concentration gradient with this one. So in active transport, we are going to use energy to force molecules from areas of low concentration to areas of high concentration. We're going against the gradient in this case. So you can see on the bottom we have a carrier protein. What other method of transport did we say use carrier proteins? What was it? What part of passive? What type of passive transport? Facilitated diffusion. Good. So yes, we have our carrier protein with our molecule. In this case, and this picture is on your, should be on your notes, so you'll want to do one thing. Circle. I think it's on there. It might not be.
On this one?
No. Might not be. Okay, well, anyway. No, it's not on there. Never mind. But one thing you will want to note with this diagram is the presence of this little spiky thing down at the bottom. Again, that is ATP, our cellular energy. So ATP is going to be important because it's going to help the transport protein to force the particles against the gradient. We need to put energy in. It would be like if I take that elevator example and I say that I'm going to shove all of you guys back into the elevator. That would be active transport. I'd be cramming you from an area of low concentration into an area of high concentration.
Wait, why?
That's just the example.
No, but like, why is it low to high? No.
Because you'll see a couple reasons why. There's a couple reasons why. Anyone need more time with this?
I bet no, it's not.
No.
Okay.
That's not funny, too.
So, first thing I want you to do with your partner, jot down a couple things. What do you notice about this picture? What do you have questions about? What do you recognize? What do you not recognize?
That is accurate.
Okay. Noah, what do we see?
Not the dots, but like this, where it's coming out of, it kind of seems like it's popping.
Okay, so this one?
Yeah,
So it looks like it's kind of popping out. Okay.
that didn't make the cut.
Sure. What else do we notice?
Cytoplasm.
Harley.
Proton.
So what's the proton? Wait, what did you say? Hypotonic. Okay, so we have, which side would be hypotonic? The red one? Why?
No, it would be the blue one because the water's going in. I don't know, out.
So in this case, we're not going to really, I'm going to cut this off here. We really won't talk about them in terms of hypotonic or hypertonic. There is, however, a lot more of this on the outside than there is on the inside. What else do we notice? Gavin, what do you notice? So we do have the plasma membrane. What is the plasma membrane doing?
Allowing certain things in and out.
It is allowing.
It kind of
But-
like a pimple
What is it actually like physically doing?
bursting,
So this one's bursting. What is this one doing?
sucking.
It's like pulling it in. It's folding around. You can see that it makes this little bubble.
Oh, I
Yeah,
get it.
that's
Yeah.
what I was saying. It was like sucking it in.
So that is something we will talk about here on the next slide. Anyone have any last minute additions? So we are doing this in the cytoplasm. This is going to happen between the inside and outside of the cell. Okay.
Exploded waste.
So in vesicular transport, so this is called vesicular transport. Where have we heard of something that sounded kind of like vesicular before? What was it? Not vascular.
Well, it sounds like it.
Or it's not working.
Something funny, guys.
Okay. So where have we heard of
Vesicular cancer.
No.
that a thing?
That's testicular.
Does that make the cut?
So, where have we heard of vesicle before?
Beans.
No.
Bess.
Anyone heard of
Bess.
a...
Besseline.
No.
Think about the cell.
The
The
cell.
mitochondria.
No.
Vacuole.
Not
Chloroplast.
close, but not quite.
Cytoplasm.
So the vesicle is a little bubble. Guys, come on.
Guys, pay attention.
The vesicle is a little bubble that is made inside of the cell. It's a storage capsule for different things. So endocytosis. What does endo mean?
Inside.
And enter.
Enter. I don't know why my marker's died.
Your markers are broken.
Yeah, I don't know why. Endocytosis. Endo means enter. Cytosis has to do with the cell. So we are entering the cell. Exocytosis. What does exo mean?
Exiting the
Exit. So we are exiting the cell. So in endocytosis, we are going to be using energy. Not that is written here, but we are using energy to bring either large substances or large amounts of substances into the cell from the outside environment. We have a couple different examples of this. You can get your giggles out now. So we have two. We have penocytosis, which is cell drinking. We are going to take the extracellular fluid
Pronounce it pine.
and we are going to kind of bring it in a large vesicle along with anything that was in there. So any small particles that were drifting around outside the cell would come in through that vesicle. So you can see in this, it's literally folding around the particles. So it's creating a little vesicle, a little bubble. The other one, phagocytosis, we are going to be eating large particles. Yes, that is how it's pronounced. So in phagocytosis, it's going to be considered cell eating. So we are going to be engulfing large particles. So this could be cell eats cell, this could be cell eats macromolecule, this could be cell eats a whole bunch of different things. Cell transport. So exocytosis then is going to be the exiting of particles from the cell. It's going to be the part where the cell expels waste. It goes to the bathroom. It lets loose a whole bunch of waste products. It could also be used to secrete different substances. For example, if we need to make a whole lot of hormones or something like that, we can use exocytosis to dump them all at once. So endocytosis is going to be enter. Exocytosis is going to be exit. Both of these are going to use energy. They're both going to also fold the cell membrane. The vesicles are able to bond to the cell membrane. So, a little bit of a critical thinking question. If the vesicles are able to bind to the cell membrane, they're able to...
That make the cut?
They're able to connect with the cell membrane. So if they can connect with the cell membrane, what do the vesicles have to be made of?
Oh,
DNA.
What type of lipid?
carbohydrate.
Phospholipids. Yes. So if the cell membrane is made of lipids, that means that the vesicles also have to be made of lipids. So you can see in this picture, maybe now it'll work. Aha, now it works. So in exocytosis, we have this vesicle you can see is made out of phospholipids. It's going to bond to the cell membrane and it's going to force everything out. Where is the high concentration of little blue particles on this side?
Out there. Up top.
The high concentration is outside. So usually we denote concentration with brackets. Where is the low concentration? Inside the cell. So we are taking, again, through active transport, the particles are going from the inside of the cell to the area of low concentration. In endocytosis, we are going to take particles from the outside. We are going to bring them in. Now, this is a type of active transport, even though it doesn't look like it, because we have to physically bend and shape the cell membrane to create that vesicle. It needs to pinch off to create that little bubble, that little vesicle. So this one is going to bring particles into the cell. Any questions on the basics of endo and exocytosis?
You said put it down.
Yes. So, this also brings me to something that we very briefly talked about when we introduced the cell membrane, and those are receptor proteins. So receptor proteins are going to be important because they receive signals from the outside of the cell. So you can see in my little diagram on the bottom, you can see the little receptor protein. And it's going to bind to this molecule, whether it's a protein, whether it's a carbohydrate, whether it's some sort of signal molecule. And it's going to trigger a response within the cell. So whether that could be creating vesicle, that could be creating hormones, creating proteins, creating a whole bunch of materials that get shuttled out of the cell. These receptor proteins are going to be important for that. They're also going to be important. You can see on the top we have receptor-mediated endocytosis. Big fancy words. Pretty much that just means that in order for this endocytosis to work, we need to trigger some of these little receptors, these little claws up here. So whenever a molecule binds to it, it will trigger a response and it will create a vesicle. So these receptor proteins are going to be very important to how the cell works and how it brings different particles into the cell. We will actually see a video of this here in a bit, if we have time for it today, about this type of endocytosis. So, thinking back to beginning of the year, which of these 7 characteristics of life did this apply to? What was it?
Oh, I was just saying trigger response.
Like reaction.
Reaction or response to stimuli. So you remember at the beginning of the year, guys over here, we had our 7 characteristics of life. One of those was response to stimuli. In this case, we are responding to stimuli. We have a receptor here, responds to this particle, this stimulus, and it elicits a response, whether that is to create a vesicle, whether that is to make a bunch of chemicals, whatever it's for. We are showing that our cells are alive because they can physically respond to stimuli. So, looking at the different parts that are involved in this endocytosis and exocytosis. So, if we start from the very beginning, we start down in the nucleus. The nucleus sends a message, a signal, a instruction to the rough and smooth ER. Does rough ER and smooth ER make a couple things? What did we say they made?
ATP.
No.
Protein.
Protein was one of them. Lipids was the other one. So the rough ER makes proteins because it has a whole bunch of ribosomes. Smooth ER, can't speak, makes lipids because it does not have ribosomes. So both of those are going to be packaged into vesicles and sent to this big stacked thingy. What was this again? What is this organelle? The Golgi apparatus. Good.
It's a mailbox.
Yes, it is the post office. It's the mail carrier. So it's going to package all of these lipids and proteins with some sort of tag. From there, it can be sent out to a different part of the cell if they need it in a different part of the cell. It can also be sent to the cell membrane.
And that's what's
And that's what's happening in exocytosis. We are taking products that have come from the Golgi. They are being sent out. They are being fused with the plasma membrane, and they're being sent out. Does that make sense? Like at this point, we aren't learning as much new stuff. We're learning applications for what we already learned. So this should all be review kind of at this point, just tying things together. And then, of course, we have our endocytosis. Particle comes in, lysosome binds to that and breaks the product, or breaks the molecule down into its bits and pieces. So the other half of this, we talked about endocytosis, we talked about exocytosis. So the other one we were missing is the ion transport. So ion pumps. Now your paper does say that this is an ion pump. Mine, I just tried to clarify and say that it was sodium and potassium. Sodium Na, potassium K, both of which are ions. In this case, this is a type of membrane pump, kind of like our facilitated diffusion, that is going to allow molecules to, again, go from low concentrations to high concentrations by pumping them with the use of ATP. So we have sodium going in. Sodium concentration normally in the side of the cell is low, but it's high outside. Potassium is is low outside and high inside. So we are pumping sodium out of the cell. We are pumping potassium into the cell. And what that's going to do is it's going to create a very, very strong electrical gradient. And the point of all this is to do things like fire your neurons. So anybody know what a neuron is?
It's in your brain.
It is a brain cell. So neurons are going to fire in your head thanks to this protein. So the ion pumps, the potassium the sodium potassium pump will pump ions to the opposite sides of the membrane where they don't want to be, where they're high concentration. What that does is it sets up an electrical gradient for our nerves to work. So when your nerve gets a signal into your head, so it transmits from one cell to the other, as soon as it triggers that cell, it's going to open a bunch of doors and flood the cell with sodium and potassium. And that's what causes, kind of like flicking on a light switch, the electricity to go through your neuron. And so every note you're writing, every thought in your head, every word you're speaking right now is controlled just by the movement of these two particles. A couple other things, but mainly the movement of these particles. So every thought you're having right now, I had a couple people have a little bit of an existential crisis with that one, but every thought is just a bunch of chemicals moving around in your head.
Probably.
Any questions about the sodium-potassium pump? And like I said, other ion pumps will work very similar to this. We have actually seen a different type of ion pump that we will talk about after the video that works very, very, very similar to this.
What are the practice questions?
We'll get to them. Any questions about this? Anyone need more time rating? Okay. So we're going to watch this video, and then we're going to go through a couple... No, it's not a song. We will go through a couple different examples with this. Okay, so first practice question. So please flip to the practice questions page. First thing we are going to do, using the vocabulary that we learned about today. So active transport, concentration gradient, diffusion a little bit, all of those. Use these concepts and explain what is happening in the diagram below. Let's take a couple minutes. Kayla, what's happening in the video or in the picture? How so? How can you tell? Okay, so they're going through the proteins. Are they going towards high concentration or low concentration? They're going towards high concentration, yes. So we have these hydrogen ions, these H+, and they're going through these different proteins from low concentration to high concentration. Now I will say that you cannot see the ATP in this. This process, I believe, uses ATP. But the main goal of it is to produce ATP. Anybody recognize this diagram?
It's the plasma membrane.
It is not the plasma
No, it's the
membrane.
mosaic.
This is actually from a previous unit.
Yeah, the mosaic.
What from the last unit? What is it? It is not photosynthesis. It is cellular respiration. What step of cellular respiration produces the most energy? Keep going. The electron transport chain. This is the electron transport chain. We have the mitochondria, has its double layers. It's going to pump hydrogen into the middle layer. It's going to move them from low concentration to high concentration. That hydrogen is used to make ATP. Tomorrow we will continue these, and then we will practice a bit with cellular respiration. Any questions?